Hi, my name is Hanna Bauer and I'm a grad student here in the College of the Coast and Environment. I'm originally from Portland, Oregon, and I work in Dr. Cassie Glaspie's lab in the Department of Oceanography and Coastal Sciences. This summer, I traveled to the Florida Keys to collect lionfish like El Diablo here for our research on marine community ecology.
I'm Dr. Cassie Glaspie, an assistant professor in the Department of Oceanography and Coastal Sciences. Lionfish are an important species to study because they're a top predator on coral reefs, and in the United States, they're an invasive species. The lionfish invasion is bad because invasive species are kind of like pests, like the weeds in your lawn. They're not supposed to be there, and it's really hard to control them once they get into your lawn, or in this case, once the fish gets into the reef. It pretty much eats everything because it is a top predator, and there's not much that can eat it in its invaded range. So pretty much humans have to focus on removing them from reefs if they want those reefs to be lionfish free.
Because as a science community we're interested in controlling invasive lionfish, my project specifically looks at lionfish living in deeper reefs. We mainly control lionfish by spearfishing them, but most divers only go to 30 meters deep. Lionfish can live down to 300 meters, so that leaves a big area where we aren't controlling the lionfish population. I use a model called a coupled foraging bioenergetic model, which sounds intimidating, but it looks at how much can a fish hunt, how much prey can it eat, and it looks at the metabolic process of the fish to see the energy in, the energy out, and how much the fish is growing. So we put those two parts together. In order to use that model, we have to measure some parameters in the lab to see how fast do these fish grow, how much do they hunt at different lights that are available at different depths. So I will have 20 fish that I will put in tanks and test how much they eat at different light intensities. So as we go deeper in the ocean water column, the light decreases and the different wavelengths of light filter out. So I'm looking at how this reduced light affects how much they can eat. We can put this into the model and then identify where in our oceans lionfish are growing the fastest and where we should target these conservation efforts. So this summer we traveled to the Florida Keys to actually collect the live fish themselves. Our fieldwork was very fun. We got to go diving with nets to collect live lionfish. Lionfish have venomous spines, so it was challenging to avoid getting pricked by these spines, but thankfully we all made it out in one piece and with 40 live fish that we then drove in a straight shot back to Baton Rouge. My favorite thing about LSU and the oceanography department specifically is how we have diverse research going on in a very collaborative environment. So there's many labs doing different things that gives everyone an opportunity to see different disciplines and how they do science, but also people are always around to help me feed my fish, to help me change the water, and it makes it a much better experience. Being able to do science on a regular basis is very exciting because I feel very involved in sort of progressing what we know about lionfish or what we know about these specific models forward, and it's also very hands-on. So you see that science can be about the daily details, but you also still have to connect it to this bigger picture that you would learn from, say, a textbook, and kind of being part of that in between is really exciting and really pushes you to learn more.